Good morning. It's October 17th, and this is your daily brief in science. Here's everything you need to know. A groundbreaking study published in the New England Journal of Medicine has revealed that HIV-positive individuals can safely receive kidneys from deceased donors who are also HIV-positive. This study analyzed 198 kidney transplants and found comparable outcomes for organs from both HIV-positive and HIV-negative donors. Participants, all HIV-positive and suffering from kidney failure, were willing to accept organs from either type of donor based on availability. The study tracked their outcomes for up to 4 years, indicating high survival rates and low organ rejection rates across both groups. Some patients did experience temporary increases in virus levels. However, these returned to undetectable levels over time. Co-author Dr. Dori Segev from NYU Langone Health emphasized the safety and positive outcomes associated with these transplants. Historically, stigma and outdated laws have discouraged HIV-positive individuals from becoming organ donors, as highlighted by sociology professor Carrie Foote. She noted that allowing HIV-positive individuals to donate organs not only benefits them, but also increases the overall availability of organs for non-HIV patients. In light of these findings, the U.S. Department of Health and Human Services has proposed rule changes to allow kidney and liver transplants from HIV-positive donors outside of research studies, which could take effect next year. Currently, over 90,000 individuals are on the waiting list for kidney transplants in the U.S., with more than 4,000 dying while waiting in 2022. The practice of using organs from HIV-positive donors began in South Africa in 2010, but was not permitted in the U.S. until 2013, following advocacy for research studies. Since 2019, when the first kidney transplant from a living HIV-positive donor to an HIV-positive recipient was performed, over 500 such transplants have occurred in the U.S. Dr. Elmi Mueller from Stellenbosch University noted that the study's findings could lead to greater fairness and equality for people living with HIV and organ transplantation. Genprex Inc. has marked a significant milestone in cancer treatment with the completion of the 0.09 mg per kilogram dose group in the Phase 1 portion of the ACCLAIM-3 clinical trial for Reqorsa-gene therapy. This clinical trial targets patients suffering from extensive stage small cell lung cancer, or ESSCLC. The trial has shown a favorable safety profile, with no dose-limiting toxicities reported in the completed dose group, which is a promising sign for the therapy's viability. Following this achievement, the Safety Review Committee has approved the escalation to the highest dose group of 0.12 mg per kilogram. Enrollment for this next phase is expected to begin shortly, with completion anticipated in the second half of 2024. Reqorsa, when combined with Tecentriq, also known as atezolizumab, has already received FDA Fast Track designation and Orphan Drug designation for the treatment of SCLC, showcasing its potential in the market. Recent data presented at the October 2023 AACR-NCI-EORTC conference demonstrated that the combination therapy significantly controls tumor growth in humanized mouse models. Notably, the studies indicated that the combination of Reqorsa and Tecentriq provided superior tumor growth control compared to either treatment alone. Initial results from the trial also showed that one patient experienced a partial remission, defined as a 30% decrease in tumor size after 2 cycles of maintenance therapy. Genprex is committed to developing innovative gene therapies for cancer and diabetes, utilizing non-viral delivery systems for its candidates. In preclinical studies, Reqorsa exhibited strong anti-tumor effects in xenografts resistant to sotorasib and demonstrated synergistic effects when combined with it. Looking to the future, Genprex has filed 2 provisional patent applications for the use of Reqorsa in treating mesothelioma and glioblastoma. Additionally, research collaborators are set to present further data on Reqorsa gene therapy at the 2024 EORTC-NCI-ACR Symposium in Barcelona, Spain, from October 23 to October 25, 2024. Researchers from Nagoya University in Japan have unveiled a groundbreaking technology for synthesizing high-purity, fully chemically synthesized mRNA. This innovative method circumvents the slow enzymatic reactions typically associated with mRNA production, drastically reducing the time required for vaccine development. The new synthesis approach effectively addresses long-standing concerns regarding the purity and speed of mRNA production. The research team introduced a novel phosphorylation reagent featuring a nitrobenzyl group, which enhances the hydrophobicity of the mRNA and improves purification processes. Utilizing reverse-phase high-performance liquid chromatography, the team successfully isolated pure RNA from impurities, ensuring the quality of the mRNA produced. In addition to linear mRNA, they also synthesized pure circular mRNA, which is resistant to degradation and provides longer-lasting medicinal effects. This advancement could enable quicker responses to viral outbreaks and emerging diseases, potentially mitigating future infections. The study's findings were published in the journal Nucleic Acids Research on October 17, 2024, underscoring the significance of this research. mRNA technology, which gained prominence during the COVID-19 pandemic, is anticipated to play a crucial role in treating genetic disorders and other emerging illnesses. Future applications of this synthesis technology may include the development of new mRNA vaccines targeting cancer antigens and genetic diseases. This breakthrough in mRNA production has the potential to revolutionize RNA drug discovery and expand the range of mRNA-based treatments available. Current mRNA derived from 5' monophosphorylated RNA poses risks of contamination and can trigger strong immune responses, which may increase the likelihood of side effects. A groundbreaking study published in the Journal of Cell Biology sheds light on the critical role of primary cilia in brain development. Researchers, led by Carolyn Ott, utilized volume electron microscopy to explore how these structures change in developing neurons, particularly focusing on granule cells in the cerebellum. The study found that as granule cells mature, they permanently disassemble their primary cilia. Observations revealed that differentiating granule cells exhibited shorter and fewer primary cilia compared to their immature counterparts, indicating a significant deconstruction process during maturation. The research team identified novel intermediates in this cilia deconstruction, suggesting that multiple pathways exist for this process, which may vary depending on physiological contexts. They specifically termed this phenomenon cilia deconstruction, to distinguish it from the transient disassembly that occurs prior to cell division. Granule cells are the most abundant neuron type in the adult brain, making them crucial for understanding the effects of primary cilia modifications on neurodevelopment. Experts believe this research opens new avenues for investigating cilia dynamics and their implications for brain health and diseases, including potential links to medulloblastoma. The study also highlights the connection between this remodeling phase and various neurodevelopmental disorders, such as autism spectrum disorders and schizophrenia. It emphasizes the roles of transcriptional control mechanisms, synaptic activity, and glial engulfment in shaping neural circuit remodeling. Recent advances in understanding the molecular processes governing these changes have been underscored, particularly through studies on invertebrate models like C. elegans and Drosophila melanogaster. During granule cell development, the ligand sonic hedgehog activates pathways that promote cell proliferation and differentiation, but cells stop responding to SHH as they differentiate. These findings suggest that a deeper understanding of the molecular mechanisms involved in synaptic remodeling could provide valuable insights into neurodevelopmental disorders and inform potential therapeutic approaches. A recent study from UC San Diego has uncovered significant biological differences in how men and women experience pain relief. This research reveals that men primarily rely on endogenous opioids, the body's natural painkillers, while women utilize non-opioid pathways for relief. The findings are particularly striking. When the opioid system was blocked in men using naloxone, their pain relief from meditation was inhibited. Conversely, this same intervention increased pain relief in women, highlighting their dependence on different biological mechanisms. These results underscore an urgent need for sex-specific pain therapies, as current treatments often fail to effectively address women's pain. Women generally respond poorly to synthetic opioids like morphine and fentanyl, leading to higher rates of dependence and addiction. Dr. Fidel Zaidan pointed out that women may require higher doses of opioids to achieve the same level of pain relief, further increasing their risk of addiction. The study involved 98 participants, including those suffering from chronic lower back pain, who underwent meditation training while their responses to naloxone were monitored. Notably, individuals with chronic pain experienced greater relief from meditation compared to healthy participants, emphasizing its potential effectiveness in pain management. Funded by the National Center for Complementary and Integrative Health and the National Center for Advancing Translational Sciences, this research was co-authored by several experts in the field. Dr. Zaidan stressed the importance of considering these sex-based differences when developing future pain treatment strategies. In summary, the study provides clear evidence that men and women utilize different biological systems for pain relief, necessitating tailored approaches in pain management. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.